of innovative activity 100 years later that the original researchers were doing still already some analysis with microbes. Okay. I started in PhD in bioremediation 30 years ago, and I'm still very excited to see how much innovative activity we can find and we can develop in combination with this wonderful crew of researchers. So I'm going to give now the stage to Eduardo Diaz, who is a member of the organization Madrid Matchday, which is a regional government association which is promoting research and innovation. And then our wonderful coordinator will give the...
Yes, thank you, Abraham. It's a pleasure, and I want to welcome you, all of you, to Madrid, because, I'm representing the regional government in the area of innovation, of science and universities, and we are, for example, promoting not only R&D but also technology transfer, innovation in general, the dissemination of science culture, even the universities, the accreditation of universities and professors. But for us, this event is very important because for us, biotechnology, and we have been working on it for a long time, is a very, very important sector in Madrid. Madrid is accounts for about 30% of Spanish startups in the area of biotech. But we know that biotech, for example, is a sector that will have a big impact in the future. Probably in 20 years, about 40% of the activity, according to international studies, will be affected by biotechnology, not only in the agrotech or on the manufacturing sectors, but bioremediation probably will be growing a lot in the next year. So that's why we think that it's very important to push this type of activities. And we are confident that the startups like Medfilter will be essential in that process. This transfer of the technology will be very, very important, and we want to support this if possible. As I said, we have about 30% of startups in biotech in Spain, not only in biotech but also in deep tech. They account for about 1 billion value. And in case that any of you want to start activities or collaboration with Madrid-based companies, corporates, partners, universities, of course, you have us to contact, and we can introduce you to anybody we can, and it will be a big pleasure. So welcome again, and thank you for the invitation.
Thank you very much. So just a few organization tips. We will have presentation, but there will be the Q&A session at the end of this workshop. So please, for the audience online, ask your question in the chat during the presentation. We try to collect them and at the end ask to the speaker, as well for the audience here. And here you have the QR code, which please, if you can scan, and there is opinion poll, because we want to check what is your awareness about the bioremediation. So displayed at the end of our workshop, but now you can also start work on that and see what do you know about the topic, and we will be appreciate if you can help us and fulfill this opinion poll. So now let me introduce the first speaker. I would like to invite Giulio Zanaroli from University of Bologna, our great coordinator of Nymphe project. So he will introduce you to the bioremediation world.
Yes, thank you. Good morning, everyone. And before starting, I would like also to welcome warmly all of you, both in presence and offline, and also to thank the main organizers, or the organizers of this first workshop from the Nymphe project on bioremediation. So again, Abram, Esteban Nunes, and Medfilter, and Innovo, and also would like to thank Madrid International Lab, who is hosting us in this very nice facility. I also would like to say that this is the first workshop on bioremediation that the Nymphe project is organizing. We will have some more in the future, because actually one of the aims of our project is not only, let's say, to do research and innovation on this field, but also, let's say, to share the new knowledge with people, both from the science, the industry, and the society, in order to favor, of course, this, let's say, exploitation also of knowledge in this field. So now I think it's time to start with the presentations, and so I will first introduce bioremediation and, in general, the approaches used or the techniques used in this field, starting, let's say, mentioning. Okay, this is, oh, now I understand, sorry, how it works. So first of all, I would only to remind that, as we all probably know, chemical pollution is really a problem, a major problem, a major ecological problems that is really widespread all around the globe due to many different human activities that are actually releasing a huge number of different chemicals in the environment. Not only those most commonly known, such as petroleum hydrocarbons or their derivatives, but also new emerging pollutants, such as pharmaceutical compounds that we actually contribute every day in releasing into the environment. But also, of course, pesticides and other chemicals used in agriculture. So all these activities and chemicals are actually affecting different environmental matrices, from soils to groundwater, surface water, even air. And of course, their accumulation is causing huge effects. Also, let's say, or may cause huge effects in terms of human health, but also health of the environment. And this is the main, let's say, starting problem, where, for example, we can observe already loss of biodiversity and of ecosystem services and functions that are sustaining the health of our environment. So actually, bioremediation, sorry, it comes with some delay. So bioremediation, what is bioremediation? Bioremediation is a process that is using living organisms, and actually mainly microorganisms, to degrade or detoxify those chemicals, those pollutants that are released in environmental matrices. And actually, bioremediation can be applied on one way to also reduce the release of chemicals to nature, for example, when treating wastewater before they are discharged in the environment, but also, of course, to degrade or remove those pollutants that have been already released and have accumulated, causing these problems. Being processes using living organisms, bioremediation typically is considered to have some benefits compared to other remediation approaches, since these bioprocesses are taking place under conditions that are life compatible. So they are taking place, for example, at ambient temperature, and do not need the use, for example, of highly reactive chemicals or solvents. For these reasons, actually, these processes typically have a very limited alteration, produce a very limited alteration of the properties of soils or anyway, the contaminated matrices that are treated, and very often also may be competitive in terms of chemical and energy consumptions. So this kind of nature-based approaches can promote not only the decontamination, the degradation of the pollutants, but in principle also the recovery of the biodiversity and the revitalization of the site after the remediation, which means, of course, favoring the recovery of ecosystem functions and services. So microbes are the main actors, not the only ones, but the main actors in bioremediation processes, as actually they are able to degrade these pollutants using different common approaches. I will not enter into the details, but just to let you understand, some microorganisms can actually use the pollutant as a growth substrate. So they grow on it, they feed on it. And while doing this, of course, they fully degrade it. Of course, in order to do that and for growing on this pollutant, they need also to consume other resources that they need for growing. For example, they need to respire oxygen if they are aerobes or other chemicals in anaerobic environments. They also need to consume nutrients, so other elements needed for their growth. Other microorganisms can degrade the pollutant while consuming a substrate, so another chemical that is sustaining their growth, still anyway having the same requirements in terms of other chemicals needed for their growth. Some others may even respire the pollutant instead of oxygen. In that case, very often we do not observe a true degradation, but only the transformation of the pollutant in another chemical that, however, could be anyway then degraded through other microbes. So these, let's say, possibilities, these capabilities are at the basis of many bioremediation processes and techniques. However, in order to be exploited, we have to intervene because very often in a contaminated site, there are many factors that are not allowing microorganisms to degrade the pollutants. And these are typically related to the lack of some elements that is needed by the microbe for growing, for example, of oxygen to respire or other, let's say, electron acceptors, the lack or the presence of unbalanced nutrients, or the presence of a growth substrate that is sustaining the degradation process, but also the lack, of course, of suitable environmental conditions in the matrix, such as pH or redox potential, or even the lack of degrading microorganisms. So what is bioremediation? What are doing bioremediation techniques? Actually, okay, sorry, again. So bioremediation techniques are engineering the ecosystem in such a way to shape the environment to steer the activity of degraders. And this means, of course, or could mean adding microbes, biologics able to degrade the pollutant if they are absent, but also controlling the environment and supplementing whatever is needed to boost their degradation activity. And this may be done, of course, directly in the contaminated site, so-called in situ approaches, but of course can be done also so-called ex situ, with so-called ex situ approaches, so in facilities where we can even better control these environmental conditions and further boost the biodegradation process, such as, for example, in the case of soils with a kind of biocomposting processes. Of course, bioremediation techniques are already, in many cases, quite effective in degrading the contaminants in pollutants, but we still, in many cases, are facing some limitations. And actually, one of the main bottlenecks of bioremediation is the complexity of the real contaminated sites and matrix. where we find the pollutant, where actually we find typically multiple pollutants which may require the activity of different degraders, and anyway also where typically the microbes able to degrade the pollutant have to interact with other living organisms that are present in the site, either large higher organisms, but also complex microbial communities that are normally inhabiting all the environments. And actually these interactions are thought to, let's say, reduce very often the competitivity and the activity of the degraders. So based on these, let's say, possible limitations, we can conclude that actually we need to consider a broader and system level in the development of bioremediation processes in order to unlock the full potential of bioremediation. And actually this is, let's say, the approach that is taken within the European project Nymphe, that is entitled New System-Driven Bioremediation of Pollutant Habitats and Environment. That is a research and innovation action project that has been funded under the topic Zero Pollution 110, and that has started 1 year ago, January 2023, funded by the European Commission with approximately 5.5 million euros. This project, let's see if it moves. I'm sorry. Okay, this project is a collaborative work that is actually carried out by a large number of partners, 18 partners, bringing very different and complementary competences. So we have 99 universities and research centers, but also we have small and medium enterprises, a large company, and 2 NGOs from 12 countries around Europe. So the main aim of Nymphe is to develop bioremediation and revitalization strategies based on bioremediation systems that consist of assembly of multiple biologics. And actually, we want to assess these bioremediation strategies from many points of view and to showcase them at 4 real contaminated sites. We will use also 1 site for using genomically edited organisms. Of course, the final aim is to remediate and revitalize, and in particular to remove at least 90% of the main pollutants from the 4 sites and improve their biodiversity status. So polluted sites are a core aspect in the Nymphe project. We have selected 4 sites that are contaminated with typical pollutants that we find all around the globe. We have an industrial site located in Italy, which has groundwater and soil contaminated with petroleum hydrocarbons and chlorinated hydrocarbons. We have an agricultural site located in Crete, Greece, where of course the soil is contaminated by pesticides, but also by microplastics that are released, for example, from the mulching plastics. Then we have a facility, a municipal wastewater treatment facility here in Alcalá, where the bioprocesses will be developed to degrade pharmaceuticals and microplastics in municipal wastewater. And finally, we have a site in Germany representing the so-called hyporheic zone, so the bed of rivers and the overlaying water, which is contaminated by pharmaceutical through the actually release of treated wastewater upstreams. We have also a 5th site located in Slovak Republic, where actually we will test in a confined way some genomically edited biologics to remediate matrices that are located in other sites. We already know about the main bottlenecks of bioremediation that Nymphe is proposing to overcome using a nature-inspired and reality-tailored system approach, consisting actually in the association, in the use of multiple biologics, but to improve the bioremediation efficiency of these degrading organisms. And finally, very important also in the development of so-called microbiome management strategies, meaning strategies that can allow us to control the behavior in complex microbial communities where these degrading microorganisms should act. The Nymphe project follows a tiered approach. The first tier consists in the selection of a set of biologics that are suited for high performance degradation of the pollutants that are characteristically occurring at the 4 sites. So we will consider enzymes, microorganisms, plants and animals. This will be the subject of other, of course, presentations. So I'm not entering into details. That will be actually combined by forming so-called multi-component systems that will assemble these different biologics in order to treat the pollutants typically occurring in the different sites. In this phase of the project, we will assess, of course, their efficiency when working together, and we will also collect the information to model this relationship between the microbes occurring in the matrix. At the second tier, the Nymphe project also wants to develop some tools and platforms to improve the activity of degraders or degrading communities working at the system level, or improving single tools, and even using, let's say, molecular engineering. Again, we will find some more information on these approaches in a later presentation. These, of course, tools will allow us to improve the degradation ability of the biologics that will be then tested back again, both at the laboratory scale, but also, okay, here we are, but also at the 4 pilot sites of the project, where we will have also the possibility, of course, to perform a much deeper assessment of these remediation approaches, both from the technical point of view, but also from the environmental and economical point of view. We will also focus a lot on the societal assessment of these technologies, which is, again, a part of the which are topics of the next presentations. So finally, Nymphe is expected to produce a number of results, in particular the set of appropriate system of biologics for typical polluted sites, but also strategies to manage the microbiomes and the microbial communities involved in bioremediation using these biologics. We will also produce tools for the improvement of biologics and the systems, also tools for assessing from an environmental and ecological point of view those technologies. So altogether, therefore, we will develop efficient bioremediation and revitalization strategies that have been fully assessed at pilot scale, and we expect, of course, to have a number of impacts, both, let's say, environmental and technological, but also economic and societal. And now I think it is time to leave the presentation, enter more into details. So thank you.
Thank you very much, Giulio. So now we go to first session, which we talk about the development of Nymphe bioremediation strategies. And let me introduce Joana Luisa Pereira from the University of Aveiro from Portugal. And Joana will talk about the Nymphe biologics.
Want me there? Okay. Okay. Good morning, everybody. Thanks for the invitation to be here and to show up a little bit more about the Nymphe biologics. Julio already introduced that one of the main aspects of Nymphe is to combine multiple biologics, and this presentation is very well. Sorry. Okay. Okay, then, so basically Nymphe focuses on the assembly of several biologics, as Julius said. So in Nymphe, we have 11 partners concentrated in characterizing and developing new biologics from this group. Come on, sorry. It's not working very well with me. So we have basically 5 types of biologics, so enzymes, plants, microorganisms, and within microorganisms we have microalgae and bacteria and consortia. Then we have animals. More specifically about each one of them. Oh, it's working. Okay, sorry. Oh, right there. More specifically about enzymes, so proteins that are able to degrade contaminants. The approach is basically start from prospecting billions of sequences of coding for enzymes that can potentially, for hydrolases and oxyreductases, that can potentially degrade the contaminants that are typically present in the matrices focused by Nymphe. In this case, in industrial soils, agricultural soils, and wastewater. So they are prospected, and there is a primary selection. Then they are those that are found to be probably the best enzymes to look more deeply into are produced, and then they are tested for their degradative capacity, so their real degradative capacity in vitro. And the idea is at the end to obtain a set of enzymes that can be combined with other biologics to enhance their capacity to degrade the target contaminants. So at this point of the project, the partners working on enzymes already reached a selection of almost 200 enzymes potentially able to degrade the target contaminants in Nymphe matrices. So these are enzymes capable of degrading hydrocarbons, antibiotics, plastics, mycotoxins, aromatics, and so on. And so all the conditions are now set to progress to the testing in vitro of their actual ability to degrade the contaminants. Considering bacteria and microbial consortia, we are focusing in one more matrix. And this is the river sediments, the hyporheic zone that Julio told you about in the previous presentation. Again, with several typical contaminants that are present in this kind of matrices. And the approach is basically to use these matrices to enrich communities of microorganisms that can use the contaminants for their metabolism and at the same time degrade these contaminants. So several different bioreactors are used with different conditions and atmospheres. We are also using microcosms and also culturing of plants to address specifically the rhizobacteria that can potentially help the plants to degrade the contaminants. Okay. After this enrichment, there is an whole procedure of culturing, isolating, and identifying the microbial consortia and the bacteria, the strains. And at this point of the project, there are already remarkable results. So after 1 year, the partners were already able to isolate and characterize several consortia and strains that are able to degrade pharmaceuticals, as you see there, several antibiotics and others. We also have already selected communities able to degrade mixtures of micropollutants. Communities also, or consortia, able to degrade plastics, plasticizers, also pesticides, petroleum hydrocarbons, and polycyclic hydrocarbons, so aromatics. Regarding specifically microalgae within the microbial biologics, here the idea is to profit from a very efficient technology in terms of energy and costs. So it is a solar power driven technology, and microalgae, or some strains, are able to indeed promote intra and extracellular degradation of contaminants in parallel to bioaccumulation and bioabsorption. So there is, of course, a challenge related to the sensitivity of the organisms, but this sensitivity can potentially be overcome by studying or by trying to find strains that are more tolerant to the contaminants we are trying to degrade. At this point, the partners already found one species at least that is able to degrade antibiotics. So we are in the good way, but several other species are being tested. Okay. For plants, plants are used in Nymphe, focusing specifically on industrial soils. So in PAHs and alkanes, and there are two approaches that are being followed. The first one is the genetic editing of plants to add them the capacity to degrade certain contaminants, and the other approach, and Julio already mentioned it, is to profit from synergisms between bacteria in the rhizosphere and the plants and their exudates. So there is a way of profiting from the inoculations of plant growth promoting strains, and at the same time fishing bacteria to the rhizosphere that can degrade some contaminants. At this point, we already confirmed the beneficial potential of some isolated degrading bacteria that are inoculated in the rhizosphere of some of the plants. Finally, the animals, and starting with bivalves. The idea with bivalves is to focus on wastewater because, of course, they are aquatic organisms, so it would be limitative to wastewater, to aquatic matrices. And we are focusing specifically on macrofoullers that need to be controlled, and so this is another use for the macrofoullers that we need to remove from industrial settings. These organisms have high filtration ability, and they are also proven to be able to accumulate specifically metals. The challenges here relate to the knowledge on their ability to accumulate organic contaminants, and as well with the logistics of working with a macroorganism, which is a bit different from the logistics of working with microorganisms. With bivalves, we can check on their sorption ability into shells. also on the accumulation on soft tissues, the coagulation of chemicals in pseudofeces and their deposition in the bed of the system, and also on the degradation by the associated microbiomes with the shell or with the soft tissues. At this point, okay. At this point, we already found the capacity for the clamshells to absorb some pharmaceuticals, and the work is ongoing to understand better the other mechanisms where bivalves can be useful. For the earthworms are specifically targeting agricultural soils contaminated with pesticides, and the idea is to profit here from their engineering abilities. So earthworms make burrows at different depths in the soil, and they eat soil as well. So they produce casts, they produce these burrows, and they can promote the aeration of soil and improve the conditions for the soil microbiome to grow and to establish. And the idea is to promote the conditions for a degradative microbiome to establish in soils that are contaminated. And so by this way, accelerate the removal of chemicals via biodegradation by the microbial community. At this point, we already found a species that is able to promote degradation, promote significantly the degradation of several pesticides. And I think that's all. I just have the final slide. Okay, thank you.
Thank you very much. Now we're going to talk about the genetics and how we can use it for the improvement of this biological, of these living creatures. So let me introduce, yes, you have to work. Let me introduce Tomás Aparicio from CNB CSIS from Spain. Yes?
Thank you. So which is the one to pass them? So thank you for the introduction. Thank you all of you for coming today. So do you hear me? Is it okay? Okay. So yeah, I work in the CNB here in Madrid in the group of Victor de Lorenzo, who unfortunately couldn't make it today because he had scheduled a trip to India. So yeah, our task in Nymphe is applying a genetic trick to improve the biologics that will be isolated by other groups of the consortia. So in our group, we are focused on the development of genetic tools for environmental bacteria. But before getting into details, this one, no? Yeah. And to frame our work, let me introduce you and present the Standard European Vector Architecture, or SIVA platform, which was an idea of Victor, started like 15 years ago. And it's basically, you can see it, but it's basically a collection of plasmids which are modular and standardized, with the idea of reconstructing complex bacterial phenotypes. Next. Next. So I think I'm not doing well. Can you put the next slide, Caroline? No, yeah. So yeah, SIVA has a standard, so it has, I will tell you to pass, right? Okay. So it has a standard, so it has, SIVA has rules like a common SIVA backbone in the plasmid, the smallest functional sequences, the elimination of restriction sites in our plasmids, and the presence of some specific and rare sites flanking the functional modules of the plasmid, as you can see in yellow, blue, and pink. So basically, what we can do with SIVA plasmid is exchange functional modules easily just by restriction cloning. Next, please. So about replication, we have all those modules. So different copy numbers, different host range, origins for gram positives, gram negatives, everything. Next, please. The same stands for antibiotic resistances, that we have many of them. Next, please. And about the functional cargoes, well, the function of the plasmid is located, we have a wide variety of activities like expression systems, metagenomics, CRISPR modules, promoter probes, and so on. So next, please. SIVA also features a very strict nomenclature with an alphanumeric code. Next, please. But so the good thing about SIVAs is that you can combine all those modules that you have seen to generate new plasmid with different activities that basically can suit any experimental design. And the SIVA platform provides a robust and comprehensive toolbox to speed up Symbio projects. Next. So in Nymphe project, with this tool in hand, the idea, okay, wait, the idea is to develop a plasmid device now that can be transmitted to complex community of bacteria and then triggers a high mutational rate on the members of the community. So if we apply a selective pressure, typically in Nymphe will be growth on some pollutants, then the hyper-diversified genomes of the community members, we will select the best performance. Next. So upon plasmid elimination, what we'll have is an evolved community of bacteria that is performing better than the naive one. Next, please. So for this device, we need 4 things. First, it must be self-transmissible, broad host range, so you can replicate in many bacteria. We need mutator genes, an expression system for the mutator genes that you can see very well, and then a self-curing mechanism to eliminate the plasmid. Next, please. So I will just outline very briefly its part. So about the number 1, we already have a SEVA-based plasmid called p-mating, which contains a very broad host range replicon, so it can replicate eventually any gram-negative bacteria, and also the conjugation machinery that we implemented in p-mating that allows the plasmid to jump from a primary donor to a wild-type receptor in a community, and then this receptor becomes conjugation proficient, so the plasmid can jump again to another member of the community, so it can widespread along the community, let's say, conquer the whole thing. Next, please. So we already have this on hand. About mutator genes, the idea is using base editors, cytidine and adenosine deaminases, basically enzymes that transform one nucleotide in another, G pairs into TA pairs and AT pairs in GC pairs. So the idea is using these 2 types of base editors to generate many kind of mutation along the whole genomes of the community members. Next. Thanks. And we also need an expression system, a suitable one, and we have selected an optogenetic system. So it's a system that responds to blue light. So here the idea is that we have this T7 polymerase, an RNA polymerase, but it's split in 2 parts, and fused to the so-called magnet domains. So the magnet, when in the darkness, the T7 polymerase is spread in 2 parts. It's not functional, but when we put blue light, can you, next, please? what happens is the magnet domains responds to blue light, they dimerize, and then the T7 polymerase is reconstituted, and then recognize the T7 promoter and express the genes downstream, in our case, the mutated genes. So in this way, we can activate the mutation, just turning on and off, on. We can inactivate just turning off the light. So this is interesting for the kind of setups that will be used in Nymphe, typically chemostats or bioreactors, in which chemical inducers are more difficult to use. Next. And the last but not the least is the self-curing mechanisms, because we want to eliminate the plasmid from the community. So we'll use a conditional replication strategy. In general, a plasmid replicates through the rep protein, which is constitutively expressed. Next, please. So the idea is putting this rep protein under the control of inducible system. Next, please. So we have a repressor that is expressed, blocks protein expression, and in this situation, the plasmid do not replicate. Next, please. If we put a chemical inducer, the rep protein will be expressed and the plasmid replicates. So when we are running the experiment, we'll put the inducer, the plasmid is replicating in the members of the community, and when the job is done, then inducer must be removed only once, and then the plasmid cannot replicate any longer and will be lost in a few generations. Next, please. So the idea is testing these 4 ideas. We are already working on that. So testing them separately and then fuse in a single device and test it at the community level. Next and last. And to summarize, so the idea is applying the same forces that nature used to evolve organisms, but in a quicker fashion to improve the bioremediation capacities of the communities that will be isolated by other members. Next and last. So thank you very much and stay tuned.
Thank you, Thomas. That was complicated. So let me introduce Felipe Corvini from School of Life Science from Switzerland. And Felipe will talk about the development of system of biologics to tackle complex contamination.
Thank you, Agnieszka. Yeah. So good afternoon to all of you. So I'm going to talk about the systems, but first I would like to start on the challenges that we meet in bioremediation, as mentioned by Giulio. And there are several challenges we also need to consider when considering the systems to be developed. So usually we work with single strains, single enzymes in bioremediation area, and also target a single pollutant. And in the case of Nymphe, we try to focus to real case scenarios, so meaning multiple contamination. That's why we also strive to use multiple biologics. We also have the issue of low concentration of the pollutant. So it means we have to pay effort to isolate or generate biocatalysts, enzymes, and microorganisms that have low mono constant or low Michaelis-Menten constant. So we also face the poor bioavailability of the micropollutant for the microorganisms. And here we also try to consider biosurfactant excreting microorganisms and also considering the rhizosphere area where there are some molecule surfactants that can also help to solubilize the pollutant. And in the case of lack of degrading activity in our systems or organisms, then we strive at isolating, evolving the microorganisms, and in some cases, edit the genome of those microorganisms. Next issue is the lack of competitiveness of microorganisms we try to reintroduce in systems. And here we also bet on the better control of the microbiome of the degrader within the community. And there are also some issues with some environmental constraints, like in the case of chlorinated aliphatic hydrocarbons, we need both reducing and oxidative conditions. And here we try to create systems or combinations that are effective in aerobic and anaerobic environments. So for all of this reason, we think that it is necessary to assemble some systems and to acclimatize them to contaminated sites. Next, please. So using the toolbox mentioned by Joanna today, so we try to assemble the different biologics in order to tackle the pollutants we have in the real matrix, and we test them directly in the real matrix. And in cases we do not have satisfying results, the idea is to come back to the isolation and also evolution of the organisms to improve the system. And once we have a good performance, then we can go for tests on site. And again, if the bioaugmentation or bioremediation attempt is not successful, we still come back to the toolbox. Next, please. So in the case of industrial sites, we have to tackle two matrices. The first one is the unsaturated soil that is polluted by PAHs and other oil pollutant, as well as chlorinated solvent. Here we make use of reactors simulating aerobic treatment, as well as use of phytoremediation mesocosm systems. So we work with enriched microbiomes targeting multiple contaminants. We work also with genomically edited and non-edited plants. We also use plant growth promoting bacteria and pollutant degrading bacteria in case of phytoremediation experiments. And we also have at hand then the consortia based on auxotrophies that can disappear, that can vanish once the pollutant is entirely removed, as well as some evolved microorganisms, as mentioned by Thomas previously. And also then the plant growth promoting bacteria and pollutant degrading bacteria in the rhizosphere, and also using enzymes that have been improved by genetic engineering, but also by supramolecular approach. So, and for the groundwater of this industrial site, groundwater and sediment, we also use the same battery of biologics that will be combined together to tackle oil and chlorinated solvent contamination. Next. So in the case of agricultural soil, we focus mainly on microplastics and pesticides in Greece. And here we have different tools that we can combine into systems like vermi-remediation, biopiles, and composting reactors. And here, two additional biologics are the earthworms in the case of vermi-remediation, and the microplastic pesticide and plasticizers degrading microbiome and strains. And other tools are also included for the formulation of these systems. Next one. So for hyporheic zone, so we consider the matrices surface water, sediment that are contaminated by micropollutants, mainly ibuprofen and guanylyl urea. And here the approach is to make use of microcosms which are operated with defined redox potentials, working with oxic and denitrifying conditions. And here we make use of isolates consortia, the consortia that have been evolved naturally or using genome editing techniques, and also bacteria that can express or overexpress enzymes with either natural status or even mutated enzymes. And in this case, we also plan to use external stimulants in order to boost the biological activity. Next one, please. So for the wastewater treatment plant or for the wastewater treatment, so we work mainly in Alcana. There is a mistake, I think. Yes, so in the wastewater treatment plant, we mainly address pharmaceuticals, so painkillers and antibiotics, as well as microplastics. And here we use two kinds of reactors, membrane bioreactor and bioelectrochemically assisted systems like Metlans and tubular BES. And here we have other tools, biologics, that are bacterial and fungal strains that have been enriched from MBR that have been operated with infinite retention time. Then we also have the use of microalgae and microalgae consortia, and also bivalves and bivalve shells for sorption of the target pollutants, as well as some supramolecularly engineered oxidase and 3D bioprinted biofilms and granules of micropollutant degrading biofilm and granules of micropollutant degrading isolates and consortia. So this is all for the system. The activities didn't start yet. We're still in the planning phase. So thank you.
Thank you very much, Felipe. So now we're going to talk about the micro modeling and management. So let me introduce Sylvia Cretu from Blossom Microbial Technologies, the Netherlands.
So, thank you very much for the invitation. It's a great opportunity for us to be in a Nymphe project and to help and to contribute to new bioremediation methods, products, procedures, anything that can work in this field. So we have heard until now. Okay, so we have heard until now about quite some a series of strategies that can be used to address problematic environmental polluted sites. This is a very complex problem, and that's why I want to put you back a little bit and go in the direction of what is in there actually, what is at the location of the polluted sites. I don't know if my pointer can be visible. Can we go one slide, please? Only one. Okay, first. Okay, so I want to send you back to the location of these polluted sites and to have a look what is there in the local community. So we have heard the words microorganism, we have heard the words biologicals that we are going to be used to degrade the pollutants. But if we go back and we look into these systems, we actually realize that we have a local community of microorganisms, what we like to call nowadays microbiome, that have a great potential in addressing these pollutants. So in this community, we have a gigantic reservoir. Okay, this is not going to work, sorry. So we have, can we go back to the first? So we have a gigantic reservoir of microorganisms that can actually do the self-cleaning of the ecosystem. This is the idea that we use to work on different processes and products that can help at this phase. So we are all familiar, or the ones that they are not, they will hear it now, that actually in 1 gram of soil, we have a gigantic number of microorganisms, mainly bacteria, and we measure in the labs something, numbers more than people on Earth. So we like to say that in 1 gram of soil, we have more bacteria than people on Earth. And this bacteria is actually very capable of producing a large series, a large panel of enzymes that can degrade lots of pollutants. And they are also in the situation when they come in contact with plants, they are capable of modulating lots of compounds, the uptake of lots of compounds in these plants. So overall, we can say that we have a local self-cleaning ecosystem that can be addressed. So, okay, so what about if we look at this local ecosystem, and then we can we can see that some of these bacteria are capable of degrading, so using the pollutants for growing. So, and then we can say, okay, this can be the cleaners of the system. And we see also that there are a couple of them with an unknown function, or they are just the ones that they sustain the local community there. And we have also other members of the groups that can be actually harmful. We are talking here about the pathogens, or we are talking about the ones that can disturb even more the structure of the system. And then we come with the, we came at Blossom, we came with the question, what about if we build a kind of control panel for these communities? And we learn how to trigger them and how to change through this control panel, how to make them active, or how to put the ones that they are not beneficial, to put them down. And by activating this control panel with some specific triggers, we like to call that a prebiotic cocktail. You can give them any name, anything that can help the beneficial ones and the cleaners to work better and to restore the system can have can be a potential target there. And in this way, by optimizing these functions of the ecosystem there, through these prebiotics, we help the restoration of the soil, and we help the removal of the pollutants. But how do we do that? This is comes very easy in words, but in practice, we all know that things are not so easy. Or it's also not easy to transfer from the laboratory to the real field. How do we do that? So just to give you a glimpse about our technology is that we take everything that is there known in the polluted side. So we have a look at the chemical composition, we have a look at the organisms that they are there, at the bacteria that I was just telling about, and we start building a model. We call it a digital microbiome model. You have to imagine that this digital microbiome model is actually a virtual representation of the whole system there. So we integrate in this model all the information that we have available from the organism, and here we come with the genome information about how they function, about their preferred sources for growing. And we introduce here in this model also information about pollutants. Sometimes it's easy. We have seen in the previous talks that we are dealing with complex classes of chemical classes of pollutants. Not always we can have one solution fits all for all these complex chemicals. We try to add this information in this digital model, and we then build a kind of virtual microchip. That's an image how we see it. We actually never saw it before because everything is in a doesn't have yet a graphical representation, a real graphical representation, but this is how we imagine that this digital microbiome will look like. And we ran a series of tests first time when we try to see if there is any correlation between what is in there on this virtual platform and the bioremediation capacity. So what would be a possible output that will come out when this chip, this virtual chip, virtual model will be deployed in a real environment. So, okay, so we have first a step which is called model fine-tuning. And this is what we are actually doing in the Nymphe project, where we are addressing the key pollutants already mentioned. And then we have in silico activated design. So this is a fantastic process that we run so in computer simulations, where we try to simulate millions of possible treatments that can be applied in this virtual model, and we try to evaluate these possible treatments that will lead to the removal of the pollutant, to the address problem. Then it comes a step of selection and validation. We like to call it like this. For the time being is partially in the laboratory stage, but we do also have field validation for some of the pollutants that they are in Nymphe project. This validation process is a little bit complex. So we have to basically to transfer what we know from the simulation, so from the modeling, and what we assume that will work. We try to convert it in a physical solution that is finally applied to the field. So the idea that is there in the last image is that, okay, so after running the possible treatments, we should be able to say, well, we can go now and we can transfer computational solutions or something that is very virtual and works very well virtually to transfer it to the field. The process of transferring, it's not always easy. Sometimes we miss components. So we have no way to, in some parts, we have no way to say, okay, this is the 100% working formula. And here we will have to rely on technologies that already were presented by Thomas, for example. So for that kind of technology that will couple our simulations with biologicals or with manipulation of the genomes of some species, in situ manipulation. So this part is still very challenging, but I would like to give you an example of a product that we have validated. So it's about the removal of dioxins. So basically, this is a very basic and very rough representation how this microbiome biome-activated treatment, what we call, so the final product that is used in the soil is validated. So we extract from the modeling part, we extract the most promising solutions, so the most promising treatments that can be applied, and we extract also the combination of the community that is there. So we know that in order to have a complete removal of the dioxin in a soil, with a certain concentration of dioxin, we will have to combine a number of bacteria and few natural existing in the system chemicals, and to add it at a location. Now, when it comes to how this is actually added to the field, we have to rely on the experience of the people that are using the field. If it's about an agricultural field, it's a story. If it's about a restoration of industrial field, is another story. We also have stories for wastewater, and then we have to rely on the wastewater plant and the capacity that is there. So the advantage of this first generation of computer-generated solutions is that we have the possibility to introduce early stage in the modeling the type of the environment where a solution is going to be applied. So, and I think that I've tried to visualize there, so to add some visuals. So this can be, in some cases, I'm talking about single applications. So one single application, for instance, of blossom product in a greenhouse environment for removal of a pesticide was enough to keep the system, so to restore the system. And we have seen that with an application of 2 years ago. So we have seen that is consistent, the soil recovered. Now, when it comes to the application in the field, then it's a little bit different, because meanwhile, so we have started a process 2 years ago, but meanwhile we were confronted with an extreme unpredicted summer last year that could not yet be have it integrated in the original modeling. This is one of the other things that we try to raise awareness. It's very important for anyone that would like to have or to practice bioremediation. It's very important to keep track and to report to the people busy with solving the problem, to report all these local variations in climate, like water availability or drought, or pests of plants that they were never observed. And with this, I would like to stop here and rather save the time for the Q&A and discussion section, because I think we have quite some amazing things to discuss in the end. Thank you very much.
Thank you, Silvia. So this presentation ends the first session when we talk about the tool strategies, techniques of bioremediation. And now we want to show you assessment, how you assess these strategies. And we now move to Slovakia. I would like to invite Hanna Horvatova from Center of Environmental Services. And Hanna will talk about the field pilot testing and techno-economic assessment of Nymphe bioremediation strategies.
Good afternoon, everyone. Thanks for introduction. In next few minutes, I would like to tell you something about field and pilot testing and set of assessment, which are included in the Nymphe project. Yes, once again. because testing and assessment of remediation technologies will happen in the forthcoming period of the project. So these activities falls within the technological work package 4, and our aims are to prepare the 5 Nymphe sites to pilot testing on off-assembled biologic systems. We already know that the Nymphe project is focused on assemblement of individual biologics. When we have some lab technology which is effective and we would like to transfer it to the real contaminated matrices, it is usually accompanied by problems which are uncovered by this transition. So our next aim is to assess the effectiveness of bioremediation technologies at the pilot scales. So the reintroduction to the real contaminated sites for ensuring the technical feasibility of the future scale-up application. But we don't want only technology which is effective in the removal of contamination. We would like to bring a technology which is also cheap, eco-friendly, and accepted by the broad public. So we will perform also a set of assessment. And finally, we will benchmark the bioremediation technologies against the standard physicochemical remediation methods to demonstrate their benefits and importance of their implementation. Next slide, please. So the Nymphe sites are crucial for the project implementation because reintroduction of assembled technologies will be carried out at these sites. So the result of previous work packages, lab scale experimentation and evolution of biologics will be implemented at these sites. During the project proposal preparation, 5 sites across Europe were selected. Julia already introduced this site, so this part will be a short recapitulation. Next slide, please. We have in our site collection, we have an industrial site situated in Italy, which is a chemical multi-company site where the soil, groundwater, and aquifer sediments are contaminated with chlorinated solvents, petroleum substances, BTXs, and metals. Next slide, please. Next site is basically the Erpe River in Germany, where 60 to 80% of water is treated wastewater from the municipal wastewater treatment plant. And hyperic zone sediments and water at these sites are contaminated with pharmaceuticals and their degradation products. Next slide. Third site is situated in Slovakia. It is a site dedicated for confined use, for confined pilots, because these technologies tested there will be based on genomically edited microorganisms. It is basically a building which was adapted to a laboratory. Next, please. A site situated here in Spain is a wastewater treatment plant in Alcalá. The institute, because it is basically in the Water Institute, treats a part of the wastewater from the university campus. And the contaminants of interest here are antibiotics, drugs, and personal care products. Next, please. And our fifth site is situated in Greece, is an agricultural site, a former greenhouse, which is now used as an end-of-cropping cycle disposal site for plastic polluted tomato plants. And here we have microplastic and pesticide contamination. Next, please. The first year of project implementation was full of processes of the site preparation, and in general what has been done was selection of the sub-area of interest, a lot of field work like excavations, drilling of boreholes and landscaping, then sampling for chemical analysis, for lab scale experimentation, and also for optimization of ecotoxicological tests. At many sites, adaptation works like building of new infrastructure and installation works has been done, and also acquisition of research infrastructure. All of this was accompanied by requesting of permissions and authorization, for example, permission for work with genomically edited biologics, and some announcements and a lot of administrative procedures. Next one, please. As I indicated, it is important to bring technology which has overall effectivity, not only technology which is effective in terms of contamination removal. And Nymphe project has a holistic approach to remediation or to bioremediation. So we included ecotoxicological assessment. It is evaluation of the quality of matrices before and after treatment, employing an innovative integrated evaluation tool. Then ecological assessment, or elaboration and testing of a simple mathematical model that forecasts the environmental impact of bioremediation, starting from the soil and water, the chemical, ecotoxicological, and microbiological evaluation of the sites to the ecological status of these ecosystems. Next, please. Then among environmental assessment, we have life cycle assessment or evaluation of the environmental burdens associated with a process by identifying and quantifying the energy, materials used, and wastes released into the environment. In risk assessment, relevant hazards that could lead to hazardous event. And finally, connected risk will be identified for the technologies in the field settings. Next, please. Among economical assessment, we have life cycle cost and cost effectiveness analysis, which aims are to choose the best technology from an economical point of view. Next, please. So societal assessment will be based on the study of the societal aspects of bioremediation, identification of the stakeholders, their involvement into discussion about current project topics, and also 2 stakeholder workshops will be organized. And finally, biosafety and regulatory constraints analysis, which is establishment of an inventory of specific biosafety concerns and how they can be tackled, identifying of applicable legislation and steps in the regulatory process that may be improved with the proposed techniques. Next, please. And to sum it up, what we would like to bring is champion bioremediation technology or technologies which are validated in real environment and proven to be effective based on the set of various assessment. Then a selection of novel biomarkers to evaluate environmental status. An advantage would be if we can maybe bring some commercial products or to approach in bringing of commercial products, new remediation services and education activities, also novel knowledge, know-how or patent. And what is really important is to achieve a higher acceptance of bioremediation by professionals, because in many countries, bioremediation is still not accepted equally to physico-chemical remediation technologies, and also positive perception of genomically edited biologics when they are used in cleaning of environment. So that's all. Thank you for your attention.
Thank you very much. So now we deep dive into environmental and ecological assessment, which will be presented by Robin Kittel from Abitec, Czech Republic.
Thank you very much for the introduction. So welcome to my presentation. Good afternoon to everyone. So my name is Robin Kitzelt and I'm from Abitec. Let me start by saying just a few words about Abitec. Abitec is a small size Czech company dealing with the development and application of biological technologies to remediate contaminated sites. We also run the laboratories focused on microbiology, chemistry, and ecotoxicity. And in this presentation, I would like to introduce you the environmental and ecological assessments of the Nymphe as bioremediation strategies. Previous presenters introduced you bioremediation in a very detailed scale. So let me just it's funny, maybe next slide, maybe. So bioremediation briefly can be defined as a process that uses living organisms. We heard about bacteria, fungi, green plants, and in Nymphe we have even included animals like earthworms and bee whales and these remediation systems. And this strategy of bioremediation is to develop the conditions under which these living organisms will degrade or transform the contaminants in the most efficient way. And the goal is to eliminate them or to transform them to the less toxic forms. Abitec is dealing with the application of the bioremediation on the contaminated site. So we treat contaminated groundwater, contaminated soil, different matrices, and the volume usually is several thousand cubic meters. So to implement the bioremediation, it consists of many each single activities, many steps. Next slide, please. So we start from site assessment, but site assessment, it's even intervention to the location. Usually we have to do some drilling works to take samples. So we usually disturb local activities. Then when we implement the bioremediation, we're starting to consume also some energy. It's about construction works. During the operation, it's even the same. Usually after some months, you find that you need something to optimize to increase your efficiency. So it is connected with some reworks and also interventions, etc., etc. So every single activity represents some environmental impact. And traditionally, we approach to, let's say, to the process beneficials is to evaluate the decreasing of the concentrations of the contaminants in treated matrices as a groundwater or soil. It could be also soil air or different. And local authorities always pay some attention to the chemicals and, let's say, supportive substances we are introducing to this environment. But that's usually everything about the environmental benefits associated with the bioremediation. So the question is this approach is sustainable? Maybe. So I brought just a simple drawing diagram how it can seem. This was taken from contaminated sites. It was in situ treatment. We had been treating there the oil hydrocarbons. The benzene was the target contaminant. So the origin of pollution was jet fuel, and it was like chemical biological technology where we were introducing some let's say, bacteria degrading oil hydrocarbons. Then we had to stimulate them by introducing some sources of the mineral nutrients. And also, because there was very strict cleanup limits, we had to reach the concentration after the remediation 0.04 milligram per kilo. We used also the surface active compound, surfactant, which was introduced to the saturated zone to wash out, to flush out the sorbed contaminants and make them available for the degrading bacteria. It was aerobic degradation, so we had also introduced there the oxygen. I'm explaining in the details to show you on this picture what was the concern regarding the environmental beneficials. So it was the residual pollution, like concentrations after the remediation, and safety of the chemicals. But if we look at the whole picture, so there are many more examples of other potential environmental impacts associated with the implementation of a bioremediation to the location. So, in the Nymphe project, we have a unique opportunity to assess every single step of bioremediation, from designing the microbiome through the process optimization to the application on the real contaminated sites. We will even collect the data about these sites and the whole risk. So leading partner is University of Bologna, and there are more partners cooperating, having this all this complex assessment. So life cycle assessment, just in briefly, so we will gather information on the technologies, types, quantifies the raw materials, energies, and what is important, it will make an estimation of environmental impacts associated with the application of the technology in comparison to the created environmental benefits. A risk assessment will gather information about relevant hazards, and connected risks will be identified for the technologies and each approach's parameters, including biologics. So relevant hazards will be identified and analyzed. Next slide. Ecological assessment will be evaluate the status before and after bioremediation in terms about the protected species and habitats in the study areas. And the last one. And last is ecotoxicological and chemical assessment of treated matrices. Maybe you can switch on the next slide. This is something a bit new in the remediation. If we talk about the contaminants and their degradation, and the goal of bioremediation is to degrade them or transform them. So not always the transformation products are not toxic. In the case of polycyclic aromatic hydrocarbons, for instance, after biological transformation, there could be byproducts which pose substantially higher toxicity or even mutagenity. Traditional approach is to do the list of some preset ad hoc, let's say defined cleanup limits, and compare it. So usually have some table of the polycyclic aromatic hydrocarbons, let's say 16 congeners, and you have to reduce the concentration. But what happened with the byproducts, which are more toxic? So in the Nymphe project, we would like to integrate also the the determination of the real toxicity. So to take the contaminated matrix and define its toxicity using some ecotoxicity tests, having sensitive bioindicators like daphnates, algae, luminescent bacteria, and plants. And together with the results of chemical analysis and some supportive methods like microbiological characteristics like respiration rate or structure of microbial consortia, integrated in one tool which will show you what was the efficiency in terms of reducing the toxicity and also restoration of the system to the more natural stage, like say, and predict also the improvement in the way of biodiversity increasing. So regarding the environmental and ecological assessment during the Nymphe project, consideration will be taken to the whole bioremediation system impact on every detailed step. So from the selection of biologics to the assembly of bioremediation systems to successful re-implementation in contaminated sites. For me, as a person who is responsible for application of remediation in the field sites, it means that there will be available many new information which will help us to design the bioremediation from the beginning in the maximum and in the more sustainable way. So I would like to finish my presentation with one sentence. So let's use the sustainable approach of the Nymphes project as a game changer for bioremediation development, planning, and implementing. Thank you for your attention.
Thank you very much. So let's stay with ecological assessment topics. And I would like to introduce Florin Alexandru from Association for Development and Environment from Romania. So Florin.
Okay, I'm Florin Alexandru. I'm from Ademed, Romania. We are doing the ecological assessment, and that's a task which very much is very much similar to the spy satellites. We are going to evaluate every moment of what is happening on the battlefield to offer the necessary information to the political decision makers and other similar decision makers to make the right decision concerning the remediated sites. So we are the spy satellite. Okay. Ademed is an association or non-profit organization and non-governmental organization which has a lot of experience in evaluation of the Natura 2000 sites. The reason why I'm saying that is that the ecological assessment is based on both science status and expert opinion. So at least in the very beginning of the ecological evaluation, everything is based on the expert opinion. This situation is very much similar to what happened in 1992 with the network of protected area in Europe, which has been established by the habitat directives. And after establishing this network and the criteria for setting them up, the European science faced a very nice challenging, that is to evaluate the ecological status of those areas and then the ecological status of what will happen after application of the management status. So our experience is very important for that. We have been a custodian of Natura 2000 sites, so we know how to do the evaluation of a site, not only from the books, but also from very down-to-earth experience. We also know how to deal with the local stakeholders, because they also have a lot of projects dealing with the local communities. So we remember that those sites are not somewhere in the space. They are placed in communities, in local communities, and there are stakeholders there. We shall know very well what they expect from us. And then we also know how to deal with the disaster resilience and risks. This is our team. And our, okay, looks like it moves by itself. Okay. Anyway, the research, our research objectives are basically 4: to provide a characterization of the target situation for the site subject to bioremediation. And this is a crucial task because we have to know, we must know where to go. So what is the status we would like to have for the testing sites after applying the bioremediation? This is important because according to that target status, we will set the parameters of the intervention. And not only that, we will set the next stages of the evaluation, because the evaluation is a continuous process. It's not one at a time. Then we have to elaborate a methodology to evaluate the ecological status before and after bioremediation. That is, as I said, a similar situation that faced the Natura 2000 in 1992. So this methodology is a brand new one. Nobody has sufficient experience to do that. And one of the main problem of this methodology is that the area of the testing sites is very small. So we have to rely on granularity, which is extremely small. Granularity means the matrix we can map the site, the testing site. Let's, of course, elaborate the ecological risk assessment method, and then a methodology for zoning on the contaminated site. As I said, because of their special nature, those contaminated sites have to be assessed not only internally, but also in proximity to decide on the status of an expanded zone. Okay, we already know the advantages of bioremediation, but for the stakeholders' vision, I would like to remind them: reduce the negative impact of pollution, conserve ecosystem, enhance biodiversity, and more circular use of resources. Here, I have to stress the second and the third bullet: conserve ecosystems and enhance biodiversity, which is again very similar to what happens to the Natura 2000 network in Europe. Then you have listed the advantages that are set apart by the Nymphe project, starting by recuperation and up to recovering of the habitat and species to good conservation status, if the case may be around the recuperated sites. Okay. For having an idea about what we are talking in terms of policy making, this is the last information about contaminated sites in Europe. There are 2.8 million sites. And remember, this is the last information from 2016 from the European Environment Agency. There are much more today, but we don't have the information. So 2.8 million sites. Who's gonna set now a company to remediate the contaminated soils? Hands up. Nobody? I will put my money in that. Nobody? So you would like to remain on the scientific research. Okay then, but remember, this is a business. So what we do will end up in business. And the cost of recuperation or remediation of one site is about, on average, 100,000 euro. Today, maybe more because of the inflation. So this is a business as well for people interested, because I'm talking to stakeholders now. This is the repartition of the contaminated sites in the European Union. They are by 10,000, you know. They are all over Europe. Remember that these are data collected in absence of a register. The register is gonna be established in 2 or 3 years from now. So the number is much bigger. Again, this is a business and a matter of policy making at the member state levels. As I said, just a part of the contaminated sites are registered. So it is plenty of business for research activity and also for business and for policymakers, of course. So we expect at least 166,000 additional sites to be in need of risk reduction measure of remediation. That is again to have a vision, to have a view what we are talking about here in this project. So again, if a cost of remediation for one single site is 100,000, then the total cost will be 16 billion. Still nobody interested in having a company? Oh, okay, one, two. Okay, so the more I explain, the better opportunities for business are there. Okay. Thank you for putting money in my company. Okay. There's a tremendous need for financing, you see. Now, coming to our business. This is the model we are going to use in evaluation of the conservation status. So the main point here is the target situation, or the good ecological status, as it was been called in the Natura 2000. This gray line is what we are targeting. We target that line. Now, the light green one is the status where our sites are. And the trends are those colored lines. What's the point of this model? The point is that we need time to know what happens after the evaluation, after the remediation. So in the case of Natura 2000, we speak about 5 to 10 years time lapse between the intervention and the next remediation or the next management plan to be elaborated. We are going to do a sort of pilot ecological evaluation after 2 or 3 years. That depends on what happens on the sites, on the testing sites. Speaking about the model itself, we have to elaborate a set of parameters, input parameters and output parameters, but this is something which belongs to the research maybe in the first place. It's not really necessary to know at this stage by the policymakers and business entrepreneurs. The interesting point about all this model is that it is very much similar to what is actually used in Natura 2000 ecological evaluation. But remember that Natura 2000 has been established in 1992, and this model came in place 20 years after that, after passing through various intermediary stages. So it's not going to be final anyway in this project, but it's going to be the ground. So it's plenty of space for research as well, for research. For the next 20 years, who is going to join our team in ADAMED? We'll have a lot of work to do in this ecological evaluation of the bioremediated sites. So we will hire. Anybody? No? Okay. That will be a lot of competition there, you know. You will see. Good. This is the time. As I said, it's 5 to 6 years to be to pass after first long-lasting results will be seen. So the key aspects, that comes somewhere in between the stakeholders' interest and scientific interest. Similar to Natura 2000, we have to first classify the values of the parameters. So there are classes. Then we have to define nomenclators, which consist in homogeneous definitions for each class. Then we will do the evaluation, we build the evaluation metrics, which is to be done through an homogeneous procedure, and then we shall work in a scarce data environment, but ready for further assessment if needed. Now, why I'm saying that? We are going to work with big data. I don't know whether you are familiar with these aspects, but everything here and everything in the evaluation is gonna explode in terms of volume. We are speaking about 3 million of sites. Who's gonna read paper by paper everything about one single site out of those 2.8 million? Nobody. That is something for artificial intelligence, for big data, for machine learning. And what is important from this point of view is that in 10 years from now, every single person, every single expert working in this field will be requested to know GIS and data management. So that's training. Another opportunity for a business, yeah? Okay. If somebody knows how to deal with big data or with GIS? No. Okay, I have a lot of clients because we do know in ADAMED how to work with that. So when the moment will come, we are waiting for you in our training sessions in 5 years from now. Okay, the GIS database format is something that is to be accessible for decision makers, that will support further scientific research and ready for big data. There are regulation issues, that's for decision makers here. There is still no regulatory instrument for soil protection at the EU level. There are large differences in national soil legislation and differences in the density of the sites in each member state. But we expect that this business will expand. So over 60% of the European soils are unhealthy. That's what we do now. We are preparing the ground for 60% of the European territory. Don't you feel proud of you? I do. Should be very proud of us. We are speaking about 60% of the Europe. Then costs over 50 billion euro per year lost because of soil degradation. Now, in 2023, European Commission published its legislative proposal for a directive on soil monitoring and resilience. So it's gonna be a regulatory network in the near future. Some ideas about the directive on soil monitoring. That sustainable soil management will be the norm in the European Union. And soil health will include ecological assessment, which is not a procedure in place, but the Nymphe pilot procedure will serve as an input to the next EU level procedures for that. Don't you still proud of you? Okay. Thank you very much.
Thank you very much. You were very convincing of this business opportunity. So let's go to our last presentation for today. I again recommend to ask questions via chat. So anyone from online audience are interested, have some questions, issues, please write in the chat. And the last presentation will be from Patrick Rudensheim from Perseus, and he will talk about the societal assessment of Nymphe bioremediation strategies. And Patrick will join us online. I hope that you will hear.
Let me see if I will speak a little bit just to check whether you can hear me or not. I've heard that the quality may not be as good, but if somebody can signal whether you can hear me, that would be okay. Let me see. Yeah, perfect. Thank you very much. Okay, so let's start. And again, also thank you to the previous speakers, first of all, for highlighting the importance, including the last speaker, highlighting the importance of this project. I think it's also a unique project in terms of the composition and really looking at it, not only the scientific, but really how do we bring these things together in a workable, acceptable, and really embedded process. So I think that's also the thing where we are very much looking forward to. So I'm with Perseus. Perseus is a service company based in Belgium. We're a small company, but focused on mainly biosafety and biotech legislation. And you've seen that there is a part of legal assessment, which we also carry out. Plus, there is a part about the societal aspects, and I think that's also the one that I would like to go a little bit more in detail in, and at the end also call on your contribution. Yeah. The reason for that, I hope you can see it, it's actually less clear on my screen, is indeed to say societal is actually complementing the technical aspects. And we have we have a lot of technical aspects that we need to cope with. We have a big challenge out there. But one of the challenges is also the fact that you can have the best technology, you can have the best achievements. At the end of the day, if this is not fitting within a concept of society, within a vision of society, and people have either questions or have not been fully involved in it, it's still a hurdle that can block the real implementation, an implementation which, as the previous speaker identified, is really needed. So we do see, and I think it's great for the consortium that they have included these aspects from the beginning. We do see these assessments, whether it's ecological, whether it's the feasibility, whether it's the business part, whether it's the societal part. We see that as a key element for the success of making added progress in the bioremediation area. Now, of course, if you think about, you have all these big words, societal assessment is one of them. And of course, it comes with a lot of different aspects. And we will try to cover a few. Yeah. Obviously, there is a stakeholder analysis, and stakeholders have already been recognized. We've also done some part in identifying stakeholders and trying to understand what their drivers are, what their interests are in this field. That, of course, relates a lot to perception, and perception you can then also reflect in communication. We're also looking at the legal aspect I already mentioned, because the legal part, to some extent, is also a reflection of what happens in society and how society at large perceives this. So there are these very different elements that we consider to be part of societal assessment. And we're also approaching it in different ways. It could be checking on documentation. We've done some literature searches already. We're looking at other projects, because obviously this is not the first project in the European Union. We're trying to build as much as possible on what has already been done and learn from their approaches and the outcomes of that. So those are aspects that we have already done, and we are entering in a new phase of, let's say, the project also after this workshop. Obviously, there's different concerns, there's different perceptions, and well, some of them are listed here. They can range from, as we identified, some concerns may have to do with safety aspects, health aspects. Of course, there's the benefits, yeah, that relate to that. It may be, how do I get involved? How do I get a say? Can I trust authorities? How is it? So there's a lot of different elements when you start to think about the societal. And mapping those are, as I already mentioned, are actually quite important, because it also shows where, if we think about communication, if we think about interacting with stakeholders, I think it can help us to identify where we can best put efforts, or what kind of involvement should we see in order to increase the chances for success. Of course, in a transparent and informed way, but still sharing that information and making sure that people can have a view on what is going on with, on one hand, their tax money, but also with a future that, as was pointed out already, would probably really benefit from some of these progresses. Clearly, we are in an area where there's a lot. On one hand, we see the support for these eco-friendly solutions, but also with some criticism. I think the question about, is it performing? Is it really delivering? I think we all realize that it's not always white or black, that there is a lot of gray in this field. Also, as pointed out in a previous talk, the fact about does bioremediation really create a full clean environment, or do you have less toxic substances present. I think those are elements which comes into that balance, and where you can see that depending on what people know about it, how good people are informed, how good that information is shared, you can see skepticism, concerns being raised, which then leads to what are the conditions for acceptance? If it's not black or white, then how gray can it be to still achieve a matter of acceptance and see the benefits of these technology? Some of the things that are already included in the project include typically with local communities. We've indeed just launched a survey, so please take care of that. There could be stakeholder meetings and there can be discussions. We've also planned now a workshop later this year at coinciding with a major conference of the European Biosafety Association. So there's different ways to try to collect and to get information and get insights on how different groups, how different professionals, but also lay people perceive these developments. So from that, on that basis, I just, as already was indicated, I would like to remind you that we have this survey which is now launched. You'll find that it's a survey with very, very, I would say, basic questions about the technology. I think it's the first survey that we'll do. We will likely have others during the continuation of the project, but this should already, it fits within some of the first findings that we had based on literature and based on previous projects. But it's really the idea that we not only invite specialists, but also just, I would say, people interested in the topic, or people not yet interested in the topic, to understand where they are and how they perceive this these kind of technologies. I realized that it was a fast and short presentation. I was also aware of the time and some of the time that was used by the previous speakers. Obviously, if there are questions, you can always contact us or the project. But I hope that it showed you how this is integrated into a real component of assessments, which go along with the technology and which we all think are essential to really provide a full kind of view on these kind of applications. So that was my presentation. I also realized that it's not very fun to have somebody just remote speaking, and believe me, we would have done this differently if we had not had to change it at the last minute, because the originally intended speaker could not make it. But again, if there's any question, I think we will be happy to address it and look forward to a further continuation of this workshop. Thank you.
Discussion. So if any of you have any comments, any questions, any insights, so this is like the time for that. So anyone from the audience inside would like to add something to this presentation? Because I see that online yes, oh, there is a question online. Wait a second. So anyone of people present here would like to say something about it? I will check because there is one question online. Yes? Yeah, it is. Thank you. Okay, so this is the question. So Julio, I don't know who could answer that. So Joanna, maybe?
Well, actually, I would say that they were not in Nymphe, maybe Philippe and also Giulio. In the planning stage, was there a consideration of insects or not? Basically, in the project, we are not considering insects.
No, actually, insects were not considered, let's say, in bioremediation, maybe not because they cannot be useful, but we were actually at the moment focusing on soils and waters, and looking at these two environmental matrices, let's say the proposed, at least, animals that also based on previous literature information were somehow most promising to be integrated in our bioremediation system for these two matrices. I mean, we cannot exclude that insects could be interesting, for example, in the degradation of plastics, for sure. We know already that, but not at the I mean, not in this project. For sure, yes, insects could be very interesting for the biodegradation or stimulating the biodegradation of other pollutants.
About the bees, I think more particular, they ask, yes, if the bees could be the
This is for the monitoring, I think. I mean, the assessment. I don't understand this second question, if it is referred to the ecological assessment. Okay, so I don't think this has been considered unless it is part. Yes, probably. Florin.
Okay, thank you. That's question for ecological evaluation, I suppose. So the ecological evaluation is based on the habitats. Habitats means plants association in the first place. So from the combination of the plant association, climate, and soil, we shall see what are the animal communities to be considered. At this very moment, we do not know whether precisely bees could be a parameters, taking into account that we did not yet practically evaluate the habitats, climate, and did the zoning system for those testing sites. But from our experience in Natura 2000, we shall focus on animals, species that are either protected or rare, or they are considered as indicator species, or they are considered something like crucial presence of crucial that the habitats and the other species do need them in order to survive, like a key species, let's say. So, of course, we will consider every insect species that could be on those sites. But I don't know at this very moment whether bees will be among them. Thank you.
Thank you very much. We don't have any more online questions, so.
I have a general question, very simple, that the student make me in the when I'm teaching, and perhaps for decision making is important. So students or people in the street see that you use animal or plants to bioremediate, but always they ask me, what can you do later with these animals or plants? So so perhaps in the future, even for communicating to society, perhaps it's not only that we are using animals and plants to bioremediate, so microbes, for example, but also what you can do later on. I mean, if you cover this piece of land with some plants, so can you eat this plant later on, or just burn, or can you make furniture with this plant because they are in contact with chemicals? So perhaps, I mean, this is a general comment, but perhaps for decision making in the future, we can even think about promoting this knowledge or this information later on. And my second question is to Thomas. Is yeah. you are trying to evolve microbes communities, but this can be applied to any community. Can you evolve? You can use the tools that you are developing to engineer or perform the evolution of any kind of community, independently whether it's complex, small, big, is from marine side or river side or soil samples. So.
Okay, it's a broad question. So there are limitations in the technology we want to develop. Of course, one of them is it's designed for bacteria. So we cannot tackle fungi or I don't know, protozoa or something like that. It's just for bacteria. And to be honest, the tool is specifically designed to gram negatives because, as you know, gram negatives and positives are very different bacteria, and the things that work in one side are very difficult to make work in the other side. So to be honest, gram negatives are the target organisms that we want to tackle. Yeah. Is that the whole question? Okay, thanks.
Maybe one answer to the first question of Manolo. There was recently circular economy, circular city Europe, considering also valorizing the biomass of plants that have been used for bioremediation, and really in the sense of circular bioeconomy.
Thank you very much.
Well, thank you for the project. It's really interesting. My question is about the concern of using genetically modified organisms, but in the specific place of the agricultural fields. Because, for example, in industrial sites, okay, it's just a bacteria that can degrade, but in agricultural specific fields, imagine that you have a bacteria that is being genetically modified and is able to degrade microplastic, for example. How, or at the end, the society is going to be a stakeholder. How are you treating with that situation in that specific place in which I see that you are is a part of the project?
Who's yeah? Who wanna ask? Julia, who you or who you point?
I mean, I can give a, let's say, general answer related on the Nymphe, let's say, approach. So, of course, first of all, not all the improved biologics will be genetically modified. So this is only one approach that the project is, let's say, exploring to obtain better performing biologics. Okay, but we are also exploring other approaches to improve their biodegradation abilities, etc., which are not, let's say, introducing genetic modifications. So this is sure an important aspect. I mean, and important implications of, let's say, introducing genetic modification for future exploitation in the open environment. So it is something we are exploring from a scientific point of view, and for sure, and then of course, let's say there will be an answer with maybe more, let's say, details on the strategies also to reduce the risks that could be associated to this approach. So this is an important aspect we have to consider. It is not the only approach we follow for improving. Of course, we will test these new or improved, also genetically edited, let's say, biologics in the project, and we will do it in experimental closed systems. There is a debate, I think, on, I mean, it is not at the moment possible to release in the open environment genetically modified organisms, and probably there is a debate ongoing on what also should be or can be defined as genetically modified microorganisms. It is a challenge for sure. I mean, it is to also to discuss about about this, because it is a complex issue. So let's say I think this project also has the opportunity to, let's say, add additional to add new information to this let's say debate, also through maybe these polls, also collecting feedbacks from different very different people and stakeholders. I don't know if you want to add something more specific, let's say, to the.
So thank you for the question. For the particular case that I explained before, the thing is that there is room to claim that the microbes that will arise from this strategy are not GMOs, you know? I mean, there is room to claim that, but still it's not clear, because basically what we want to do is to evolve quickly. So evolution happens in nature, you know, genomic change happens, and this is the way the organisms, you know, gain new activity. So we want to do the same and leave no trace of genetic manipulation, even though there will be edited organisms, because they probably will incorporate mutations. We can say that this already happens in nature. We are not putting heterologous genes on that organisms, just foster evolution. But we have a debate inside the project about this, and some conversation between members and with authorities and regulatory bodies to really clarify whether this GMOs applies to what we're doing or not, and it's not clear yet.
Anyone would like to add something? If not, I think time for well-deserved lunch. So I don't know, Abraham, would you like?
Yes, I wanted to thank all of you for coming today. I think after such inspiring food for thoughts, we also need food for our bodies. So you are all very welcome to join and biodegrade tapas and drinks for 1 hour. And those who are here on site, of course, for those who are online, just prepare a sandwich, okay? Thanks for coming, and big rounds of applause for the speakers, I guess.
Thank you.